And now I'd like to introduce today's speaker, NAF Medical Research Advisory Board Research Director, Dr. Harry Orr, who likes to go by Harry. Harry directs the Institute for Translational Neuroscience and is the Tulloch Professor of Genetic Pathology at the University of Minnesota Medical School. Harry is known as the researcher who found the first gene for ataxia, along with Dr. Huda Zogby. Welcome, Harry.
Thank you, Lori, and welcome to all the attendees. Should I go ahead and get started? Is it my turn now?
It's your turn.
Okie doke. As you'll see from the title slide that is on your screen, hopefully, this was supposed to be in Denver, and I was really looking forward to meeting a lot of people in Denver. Unfortunately, things have gone in a very weird direction, and we're doing this now as a webinar. We've carved out a fair amount of time for questions, so hopefully we will be able to get to all of your questions towards the end of this webinar. So first, I have to disclose some potential conflicts. Our research is supported by grants from the National Institutes of Health as well as private donations. And I also have research collaborations with several pharmaceutical companies, for example, Ionis and Biogen, Sidon2 and Execure, but I do not have any stock in any of these companies. And I guess the way the market's going these days, that's not a bad thing. Okay, so I want to first review with you the types of research grants that NAF seeked applications for this year and have funded. The research seed grants at the top, these are meant to be grants to get investigators started in new directions, to get initial or what we call preliminary data so that they can then approach and get larger grants from respective national funding agencies, for example, in the United States, the National Institutes of Health. And then the next three categories address what is one of the major missions NAF has in terms of its research program, and that is to attract young investigators into a career as ataxia investigators, both clinically as well as from the basic research standpoint. For those of you who can see my image on the screen, you'll see that I am not one of these young investigators, and it's really important that we get the bright young people into the ataxia field. So we have the Young Investigator Award, which is kind of a transition from training as a postdoc to a beginning faculty member, as an assistant professor, and then the Trainee Award, the postdoc fellowship, followed by another type of Young Investigator Award due to funds that NAF obtained from an anonymous donor, specifically targeted to young investigators interested in one of the spinocerebellar ataxias. And then the last award, you'll note, the largest one, $100,000 Pioneer Award. Again, this is driven by a specific donor with an interest in SCA3 or Machado-Joseph disease. And the feature of the Pioneer Award is it's typically for a more senior established investigator, and it really is targeting research that takes ataxia work from the basic research lab towards the clinic, which we call translational research. So just to review the process by which we go from a submitted application to a funded grant. This year, NAF received in these categories 59 applications, and we assigned 2 to 3 reviewers for each application. And these reviewers come from the Medical Research Advisory Board as well as from the general ataxia community and neuroscience and neurodegenerative investigators too, working on other diseases. The grand total this year was that we got over 160 reviews for these applications from over 50 different reviewers. So a lot of work went into the review of these applications, and it's really important to note that these were done by people pro bono. They realized how important it is fund these research grants and importantly to fund the best science that comes to National Ataxia Foundation. So here's the sort of summary. This year we funded 25 investigators for a grand total of $940,000. And I should tell you at this point, if you go to ataxia.org and /blog or just ataxia.org, and I think it's in the upper right-hand corner, hit on the blog panel, you can get a lay summary for each of these grants if you're interested in a little more in-depth description of the science that was funded this year. So in the seed grant category, we got 22 applications and funded 9. In the young investigator, 7 applications, funding 3. And down at the postdoc level, we got 9 applications and I believe we funded 5. And then the SCA young investigator, again, 14 applications, funding 7. And then the Pioneer Award, we got 7 applications, but only one of these kind of met the high standards that we set at NAF for funding these awards. And so we're funding roughly around 50% of the total application pool that we received. Now, the second aspect of the title of this talk was talking about the fact that ataxia research has come full circle, and I want to shift into that area now. And when we think of human research and specifically genetic research, research on genetic diseases, we kind of portray things in a circle. And if we go up to the top of this circle here, we start with you, the patients and the families, characterizing the disease, characterizing the genetic inheritance pattern of the disease gene in the families, and importantly, characterizing the disease characteristics in the family. What makes up the specific diseases? And then once we have identified the gene, we now move into what we call the basic science. And we in here into the research laboratory. One of the prominent mechanisms that we use to further understand the disease mechanism is to generate animal models of the disease by taking the mutant gene that causes the disease in the patient population and transferring it to these research laboratory animals. It allows us to ask very critical questions about how the disease works, and such that we can get insight into what might be a very important therapeutic target and begin to develop therapies addressing these different targets for the different diseases. And as we move up here on the left-hand side and begin to characterize these potential therapies, we are now moving into what we call the preclinical stage, which then sets the stage, if you will, for subsequent taking of these potential therapies into human research, i.e. clinical trials. And then that's what we mean by going full circle, is that we start with you, the patients and the families, go around this pathway and eventually bring back to the families a potential therapy for testing in a clinical trial. And the exciting thing in the ataxia world right now is illustrated or summarized here, is that for several of the genetic forms of ataxia listed, for some examples listed on this slide, is that we are well along this road towards the clinical trials, which the first one being a safety trial, so-called phase 1, and then the next phase 2 and 3, where we're really trying to assess in a patient population the therapeutic efficacy of our potential therapy. It's really important to understand that clinical trials are in essence research, and they are research using human patients to study the safety and efficacy of a therapy, in our case, ataxia. And one of the important mechanisms that we're exploring for some of these dominant SEAs is the use of a drug called an antisense oligonucleotide, which actually suppresses or reduces expression of the gene that causes the disease in these particular forms of SEA or ataxia. You can see at the top here, in the case of SEA2, the process is moved through the preclinical into what's called the IND enabling stage, i.e. an investigative new drug. Once these studies are done, you go to the FDA and they give you approval for moving into the actual human clinical trials, initially phase 1. And the SCA drug is right on the cusp of going into a clinical trial. And if it hasn't already started, my understand it is due to start very, very soon. And then, as you can see, SCA3, SCA1, the disease that we work on, and then subsequently SCA6 and 7 are somewhat behind SCA2. The important message is that this movement towards clinical trials for these SCAs rests upon the basic research that was done by investigators in all of these diseases, as a considerable proportion of which was supported by the research grant program funded by the National Ataxia Foundation. So we're moving from the lab to patients, and that means we've got to get ready to do clinical trials. And so this is now, I want to just very briefly outline some of the programs that NAF is partnering with other agencies, including various pharmaceutical companies, to indeed get the whole mechanism in place so that we can do these clinical trials. And the critical thing is that ataxias are a relatively rare disease, and therefore these trials are going to have to be done at multiple sites around the country, and in most cases, not just in the US, but worldwide. So to do this, the NAF is partnering with various programs in supporting these clinical trial readiness initiatives. The CRC SEA, the ReadySCA, funded by NIH in addition to NAF, and then Global SCA, which now moves the SEA clinical trial readiness to Europe as well as South America, and then Global ARCA for the autosomal recessive cerebellar ataxias. And the point here is that these studies are to get the clinical infrastructure in place so that the trials are done using in the same processes and standardized assessments and so forth from place to place, enabling us to get meaningful and critical information on the therapeutic efficacy of the drugs under study. The next thing I want to mention is another initiative started this year by the National Ataxia Foundation, and this is the Centralized Ataxia Genomics Core, or the CAGC. And the main goal of this initiative is to see if we, by using genomic sequence data, if we can increase the diagnosis of what are called the sporadic ataxias. And as probably many of you are aware, these sporadic ataxias actually represent a substantial proportion of ataxia patients that go into the clinic. And it's been estimated that this is perhaps as many as 50% of the ataxia patients. And the hypothesis that we're testing is that even though these are called sporadic, which means the underlying mechanism or etiology is unclear, we're hypothesizing this is due to a rare mutation that gives rather complex genetics, if you will, to the ataxias, making it hard to discern that they truly are due to genetic or DNA mutations. And so what the Centralized Ataxia Genomics Core is going to do is provide a repository for ataxia patient DNA sequences, as well as develop pipelines of analysis of this DNA so that it can be screened to see if there are any potential mutations in each patient that might contribute to or be the cause of their ataxia. And so what's the basis for this hypothesis? And in the case of ataxia, it goes back to a study that was done last year by Henry Holden in Great Britain. And he did essentially the same analysis on a group of late onset sporadic ataxias. And what he found was in 22% of these patients, there was an unstable AAGGG repeat. And when this repeat expanded, it resulted in a mutation that caused ataxia in these individuals. And the important thing is that this mutation was present in over 20% or a fifth of his sporadic ataxia population. And we're hypothesizing that there are similar other mutations that will be represented to a substantial degree in various sporadic ataxia populations. So I want to close by just summarizing the overall research support by the National Ataxia Foundation. So in addition to the research grants, $940,000, $30,000 was awarded to Dr. Laura Ranum to support the Brain Donation Bank. This is a very critical tissue resource which is used by researchers to test their hypothesis developed in animal or cellular models in human tissue, giving validity, if you will, to their experimental data. There's the Ataxia Genomics Core, which we've discussed, and then there's the CRC, SEA, and the other programs to develop the clinical trial readiness initiatives underway. And so the total research support in 2019 was a little over $1 million. Again, congratulations to NAF, and particularly thanks to all of the donors. I want to bring attention to some of the staff members in the NAF office. Kelsey and Marianne are really responsible for the day-to-day organization of the research program and the review of the grants and communicating with investigators who are interested in submitting applications for grants to the NAF, and helping me very much with the review process. I'm sure many of you have met Sue Hagen, who's a patient services director, and she really kind of oversees all of us, and particularly oversees me and keeps me out of getting into trouble and doing things perhaps I shouldn't be doing. And importantly, she is the link between the patients and the researchers. And lastly, on the right-hand side is Joel, who is the development director. And I think it goes without saying that all of the research efforts underway by the National Ataxia Foundation cannot be accomplished without the donors' support. And it's Joel's job to help and get the donors engaged in what's going on and stimulating interest. I'll close with just one final little point about Joel, and then move to thanking you. And again, it is an extreme honor to be part of the ataxia community. It's a terrific group of individuals. I get a lot of inspiration from all of you, and I thank you very much. And I'm glad to answer any questions that you may have.
Thank you, Harry, for sharing all that helpful information today. And just a reminder, all of you can help with the research efforts that Harry's talked about by considering joining the Core's registry if you haven't already done so. So now we're going to begin answering the questions submitted during today's presentation. And as a reminder, you can still submit questions through your control panel. If you would rather ask a question verbally, please raise your hand using the hand raise tool, and we'll do our best to answer as many of your questions as time allows today. Harry, is there a question you'd like to start off with answering?
I just certainly would like to reemphasize your point about joining the Ataxia registry, and keep aligned and keep engaged with the NAF about what clinical trials are underway and how you can participate in these trials, which ones are appropriate depending on the type of ataxia you have. I know we've asked a lot of you in the past, a lot from you in the past, and we're coming up again with another big ask, and that is, if it's appropriate for you, please consider participating in these clinical trials.
That's an excellent point.
Yes.
Absolutely. To register, for those that are interested in registering in the CORDS registry, if you go to ataxia.org and then select enroll in the patient registry at the top of the homepage, you'll be able to find the CORDS registry going that route. Also, please check the research section of NAF's website often, as information about recruiting studies and trials is updated often there. All right. Did you want to start out with a question that's been typed and assigned to you, Harry, or would you like to do a live question?
Either way, whatever. We better go with a typed question.
Actually, Becky Baldwin typed in her question. She said, why is SCA8 never mentioned in any webinar?
I think that is a really good question. Obviously, that's a disease that Laura Ranum is working very hard on. And I think the simple question right now, the straight answer to that question is that unlike the SEAs that I mentioned in the clinical trial slide, there's still some lack of clarity of what is the underlying cause of SCA8. Is it the RNA? Is it the RAN peptides generated from the mutant gene, mutant RNA? I think there's still some effort to try to understand the underlying pathological basis, pathogenic basis. What I was trying to do in that slide was present those SEAs that are clearly on the clinical trial track. And I think the important thing is that as those that are demonstrate some efficacy and success, the field will very rapidly move to the other SCAs like SCA8 and so forth. But right now, we're trying to focus on those disorders that we know clearly have a good idea of a therapeutic target, and the patient population is large enough that we can come up with a good, well-designed clinical trial to test safety and efficacy of a drug like an ASO. And once they're shown to be safe and efficacious in these trials, that'll make it easier to move to the more rare forms of SCA, like SCA8.
All right, it looks like Charles has a question. Charles, what is your question?
Is there any plan for research on ARCA1, a recessive type?
Well, certainly, first of all, in terms of the grant program that NAF supports, first of all, somebody has to submit an application in that particular disorder. And the NAF is very engaged in supporting the autosomal recessive ataxias, and will encourage, as best we can, to encourage investigators to submit applications. But I think in terms of the clinical aspects of the autosomal recessive ataxias, there is the ARCA Global Initiative, and it's designed to set up the same sort of clinical trial readiness network among the investigators for the autosomal recessive ataxia, so as potential therapeutics come along, we have the natural history data, which is something the pharma companies are very interested in terms of engaging into a clinical trial, as well as a well-organized group of investigators to apply potential therapies. So that's the best I can tell you at this time, Carl. If you know of some investigators out there, please encourage them to submit applications to NAF.
Thanks, Charles. All right. Did you want to go to a typed question?
Sure.
All right. Keith writes, is there any support available to help pay for genetic testing?
Oh,
so we
very
know
good question.
Trials we would be eligible for.
Yeah, and that kind of relates to not only the identified genetic forms of ataxia, but it also very directly relates to the genomics core initiative that we were talking about. In some places, the insurance companies will pay for DNA sequence analysis and as part of the workup and diagnostic workup for ataxia. But I think as we move forward, it's very clear that that's not going to be the case for everybody. And one of the things we're trying to do as part of these clinical consortia initiatives is to develop resources, funds that can then be used to help support these genetic tests. So I think the best thing I can tell you, Keith, is to be sure that your ataxia investigator is or physician is some way linked to one of these consortia. So as these resources become available, they can you can have access to them. And I think this is an important aspect as we move from the basic research lab to doing clinical trial and human research, is having resources available to help support patients in areas such as doing genetic testing. And that's something the Medical Research Advisory Board is working with the board of NAF to try to develop a strategy to provide such support where it's needed.
All right. Pamela is asking if there's any research ongoing for acquired ataxia, such as ataxia resulting from a stroke.
Yeah, I think the types of research going on that potentially might be applicable to acquired forms of ataxia, such as what you just described, is the research that's going on to understand the underlying electrophysiological alterations that occur in neurons that lead to ataxia. And an effort to try to develop such therapies that might bridge across many of the ataxias, because they're targeting, if you will, the underlying abnormality of the neurons that are affected. And hopefully some of these therapies will have direct application not only to broadly across the genetic ataxias, but also towards the acquired ataxias due to things like stroke.
Great. Melissa asked that she's currently unfamiliar with ASO therapy, but she's interested in learning more, especially for SCA2. So you mentioned that you believe it will be moving into clinical trial for SCA2 soon. Can you point her in the right direction for gathering more information?
I think the best thing to do would be to contact Sue Hagen at NAF, and she can put her in contact with one of the lead investigators in the SEA2 clinical initiative and see if she meets the criteria for the initial clinical trials that are going on with the ASO for SEA2. I think we're sort of, one of the very frustrating things is that these first trials will be fairly specific in terms of the type of patients that will be sought to enter these clinical trials. Understand the goal is to test the efficacy of the potential therapy in as short a time as possible, such that if it's proven to be efficacious, it can then be moved to the broad population of that specific ataxia, so SEA2, for example. And that means that typically these clinical trials will start with individuals who are relatively mildly affected, because all the research data says that the earlier we intervene, the more robust and rapid will be the therapeutic response. But once a drug is approved by FDA for SEA2, it would then be available to the general SEA2 population, and likewise for all of the other SEAs that we've talked about as their therapies are approved by FDA. So as frustrating as it is to not be part of the initial clinical trials, just keep in mind that the goal is to get these trials done as quick as possible.
Great. Lori asked if clinical trials extend to Canada.
Certainly. I know there's a very active group at UBC, I believe, and I don't know specifically other institutions in Canada, but I mean, the ARCA Global and the SEA Global would certainly include Canada.
And Linda would like to know, when do you think the SEA1 clinical trial will start with patients?
Oh boy, yeah. The hope is by the end of this year. By the end, I do know that there are a couple of lead compounds that are being moved by Biogen and Ionis through the IND enabling studies, and it's my hope is, my understanding is that at least by the fourth quarter of 2020.
Well, that's exciting.
Yeah, I think it's incredibly exciting.
Absolutely. Linda would like to know what's going on with CRISPR.
What's going on with CRISPR? Well, so what Linda is asking us to do is move into the area of therapeutics called genome editing. And CRISPR is a system or process that allows us to alter the DNA within cells within an individual. And in the case of the ataxias and other diseases, the hope is to use CRISPR to remove the mutation or the mutant gene, and therefore eliminate the need for a therapy, because the cause of these genetic forms of ataxia, particularly the SCAs, are not there. Now, there is some very exciting results. I know CRISPR has been being moved into humans for some cancer-based therapies. The major goal for the SCAs, particularly those that are due to repeat expansions, would be to remove the expanded repeat from the mutant gene, for example. And there are a lot of research programs underway. This is relatively I don't want to say straightforward, but it's quite doable. The problem the field is having is that in addition to having the desired effect on the DNA, CRISPR also hits other regions of the genome or other regions of the DNA in addition to the target that we're asking it to destroy. And it's trying to reduce these off-target effects that is the major obstacle at this point in time, as I understand it, for this general process to move towards a potential therapy in clinical trials.
Thanks, Harry. Stephanie would like to know, does the SCA readiness for type 1 and 3 have enough people enrolled to complete the 5-year study, sufficient enough for a pharma company to utilize its resources to provide a treatment for a clinical human trial? If so, will the clinical trial start as soon as year 5 or more?
Yeah, my understanding, and I'm not a physician, so, and I don't have my finger right on the pulse. My understanding is that they are still a little bit behind in recruiting individuals to be part of this study. So I encourage people in the SCA 1, 2, and 3, and I think also 6 and 7 to seek out opportunities to participate in ReadySCA or CRC-SCA. And those of you on the autosomal recessive side of the fence, if you will, to seek out investigators in the ARCA-SCA consortium for participating in these various studies and trials. A very important study to participate in is what we call the natural history. It's very important going into a clinical trial to understand as best as we can the typical course of a given disease. And it's that yardstick that a therapy will be used to assess its efficaciousness and is it changing the course of the disease? And so the more we understand about how a disease progresses in patients, the better we can design a clinical trial. So I cannot overemphasize how important it is for patients with these various forms of ataxia that are moving towards clinical trials to participate in the natural history studies. But I think we still, and my understanding is they're still looking for more participants.
Absolutely. And Sue Hagan, our Patient Services and Research Director, would like to let us all know that ARCA-1 is one of the ataxias that an upcoming clinical trial will be recruiting for. So very important for ARCA-1 families to enroll in the CORDS registry so research participants can be contacted. Michael would like to know what physical limitations there may be in participating in a trial.
I'm not quite sure what he means by physical limitations. I think the best answer I can give is that at this point in time, and we kind of touched upon it earlier, individuals who are well into the progression of their disease likely will not be included in the initial clinical trials because, again, we're trying to capture efficacy in as short a period as time as possible. So in my mind, that would be one physical limitation. I don't know if he's talking about distance from a trial center, back to the idea of coming up with resources to support genetic testing. We also are very actively trying to engage fundraising efforts to provide funds so that patients can travel from their site of residence to the nearest site where a clinical trial is underway.
Thank you. If anybody would like to ask their questions verbally, please use the hand raise tool in your control panel, and I'll call from you from, I'll unmute you and call from you from those that do use that feature. Darlene would like to know, is there research going on as to what causes uncontrolled muscle movement?
Oh, yeah. I'm afraid I'm not really, that's an area that's way out of my area of expertise. I do know that there is research underway for various aspects of abnormal movement control, such as the dystonias and so forth. And one region of the brain, the cerebellum, that's important for many of the ataxias, seems also to have a role for the abnormal movements that fit into the dystonic or dystonia category. And one area that I know that people are working on is the use of deep brain stimulation as a potential therapy for dystonia. But that's about the best I can do in terms of research and what's going on in terms of abnormal control, motor control.
Lynn says she has 2 daughters who are at risk of SCA1, but were too young to be eligible to participate in any studies. If there is a shortage of participants, would age be something that is looked at in the exclusion criteria of clinical trials?
Yes, there is a younger age. It's in the 20s, I believe. I think part of the reason is that, you know, that these therapies may impact the role these genes have on development of the nervous system, and we're just again unsure as to how young we can go with these experimental therapies and not cause unwanted side effects that, heaven forbid, would make things worse rather than better. I think the good news is that if we do get therapies that are proven to be efficacious, they will likely be approved for people who are preclinical and showing no manifestations of the disease. In which case, getting a genetic test to see if you have the mutation and when you can start the therapy. So I think that hopefully that your daughters will indeed benefit from some of the work that's going on right now in the SCA1 field, and eventually will be called upon to participate. And the more we understand about the therapies and its effects and its both on the positive side as well as the negative side, I think the age of participants will again be adjusted accordingly.
Pamela, did you have a question?
I do. Can you hear me?
Yes, loud and clear.
I can hear you very well.
Thanks, doctor. My question is this: if we are registered in CORDS, when appropriate opportunities come up, will they sort through the database and reach out to us, or do we have to be constantly vigilantly watching for trials for which we might qualify?
Yeah, that's a really good question. I know initially they will go through the registry and seek patients there. I just think that it doesn't hurt for you to pay attention to what's going on, and if you think you might fit the criteria for a trial, to seek out and ask directly, because no system is perfect, if you know what I mean, and sometimes people fall through the cracks unintentionally.
Thank you.
Sue Hagen again is the best person to keep in contact with.
Eric has a question. Eric, what is your question?
No, I don't. I'm sorry. I don't.
Oh, you're good. All right. There is a person wanting to get an update on the Biohaven clinical trial, if that is where that is going, given the state of the world right now.
Up-to-date information can be gotten again from Sue Hagen, putting this individual in contact with the right people. But I understand that the Biohaven trial is still going on. There are issues with the current problems with travel, getting people to the clinical trial sites. But they're working hard on analyzing the data to demonstrate efficacy for the trial is how I understand the process. And certainly the Biohaven therapy is one of these therapies that bridges across the different ataxias. And so if it does prove to be efficacious, the exciting thing is that it likely will impact multiple forms of ataxia.
Did text that the Biohaven trial indeed continues, and they're going to be working with their trial participants about that. Looks like Jeremy Schmaman is on the call here, and
hello, Jeremy.
Hang on just a second. I got to go and unmute him. Are you there, Jeremy?
I am. Hi, can you hear me?
I can. Welcome.
Hi, great job, Harry.
Oh, thank you, Jeremy. It's always nice to get compliments from somebody I respect like you.
No, it's just awesome job, phenomenal. We're lucky to have you. Just a word on the Biohaven story. We're not seeing patients live, but we're seeing them through either Zoom or WebEx or whatever else, whatever video we can, or just by telephone. And they're going to be assessed by questionnaires rather than the physical exam. So the Biohaven has asked us not to do physical, like the functional SARA, but we are going to be assessing people from the perspective of their own symptoms. So the scale of their, that gives a sense of what their symptoms are, how they're doing, making sure there are no side effects. That's all going to be taken care of by the research coordinators and the PIs over the phone or video. And the drugs will still be either drug or placebo. The medication will still be dispensed as before, but they're coming up with ways to make this work in a more smooth fashion, sent to people, not given at the time of the visit. And they're not starting any new subjects for the study now, because for that you have to have an in-person visit, which we're postponing until this is all over. So we don't know how long this is going on for, but the people who are in the trial now will continue. And if there's a need to extend the final visit at 48 weeks to beyond that to a later point in time, they will do that. And Biohaven is aware of the issues and working very well with all the sites to make it happen.
So the idea is to continue along as best as we can, given the circumstances we find ourselves in the middle of.
Correct.
Thank you, Dr. Schmaman. That was very helpful. Andrew Rosen, he would like to ask you if you could comment on the trends that you're seeing in research, like what are research focused on now that maybe they weren't looking at 10 or 20 years ago?
Yeah, that's it's certainly 10 or 20 years ago, a potential clinical trial and efficacious therapy was, or treatment was a pipe dream. And now, you know, the old, we can see the light at the end of the tunnel. And I think the other exciting thing is that we mentioned it earlier, the issues like CRISPR and so forth, that some of these new approaches to impact the sequence of the DNA and so forth are very exciting in the realm of gene therapy, for perhaps potentially for some of the autosomal recessive ataxias where we're trying to put something back in. There's really some exciting work going on in developing gene therapy vectors, getting them to distribute themselves throughout the nervous system, and importantly, to be regulatable. So, like I said, a clinical trial is an experiment. And one of the nice things about your standard drug is if they prove not to be efficacious or, heaven forbid, prove to be deleterious, we can stop the treatment, stop giving the drug. And by having a regulatable version of gene therapy, if we run into a similar sort of situation, then this gives us a exit, if you will. A way of stopping the effect of the treatment by having a regulatable gene therapy. So those would be the areas that I think are, in terms of the ataxias are, I find the most exciting.
Great. A couple of questions regarding sporadic ataxia. William would like to know if there's any clinical trials for sporadic ataxia. And Michael would like to know if you have had genome testing for sporadic ataxia that's negative. Is there further testing that can be done for sporadic ataxia?
Well, I would have to, for the latter question, I'd have to know what exactly was done. And I think the best thing would be to have your records gone over by, say, a genetic counselor in an ataxia clinic to see if the testing that was done is includes everything that we know. In terms of clinical trials for the sporadic ataxias, again, if Jeremy is still available, he would be a much better person to answer. But I think therapies like what's going on in the Biohaven trial, the ones that we call symptomatically targeting therapies, have a potential to be effective against sporadic ataxias as well.
Jeremy, if you're still there, did you want to comment further on that?
Yeah, hi. That's exactly right, Harry. The drugs that we have available now are for the symptomatic treatment of ataxia, with the hope that by influencing the function of the nerve cells in real time now, there may potentially be some neuroprotective effect down the road. But that could be available for anybody who's had other causes of ataxia, and sometimes the medicines we have work and sometimes they don't. But that will intersect with the approaches that you explained so nicely about the disease modifying agents for each individual ataxia. The ones that we have available, whether people are using riluzole or the new agents, troriluzole, or the one coming out that Cadent has, or the old-fashioned medications, those are all symptom-based to try and make somebody better in real time now, as opposed to fixing the underlying problem.
Thanks, Jeremy. And this may be something that, question that you both can comment on as well, because everybody is talking about it. There's a question about how does the coronavirus affect people with ataxia, and how do you think it will affect research efforts?
Well, I think that, again, Jeremy's much more qualified to address this, but just under the general rubric, I think most people are aware that patients who have to deal with ataxia have to be concerned about respiratory and breathing complications in general. And so any sort of respiratory infection like the disease COVID-19 or even influenza is something to be avoided by all costs, if you will, by the ataxia community. And this was the underlying issue we faced in having the NAF meetings in Denver a couple of weeks ago. And I think the group headed by Jeremy was very courageous in coming to the very difficult situation that the potential risks far outweighed any benefit. So the better your general health is, the better you can deal with the ataxia. And certainly the breathing and respiration is a critical area to maintain as healthy as possible.
Great. And this question, I appreciate because it certainly speaks to our global reach. Mariah asked, do we offer grants, research grants, to non-US researchers?
Absolutely. I would guess in this last pot of applications, something on the order of 25% of the funded investigators were from overseas, from the perspective of the United States overseas. Certainly in the field of SCA3 and Machado-Joseph's disease, we get a lot of outstanding applications from Portugal, where that disease is much more prevalent than elsewhere.
And William Sweeney would like to know how NAF is working with clinicians and pharma to help move research forward.
I'm sorry, I got the beginning and the end of that question, but not the middle. What's
Absolutely.
the question?
Would you comment on how NAF is working with clinicians and pharma to help move research forward?
Well, I think the most exciting thing is the effort being spearheaded by NAF to form this consortium that largely would be funded by various pharmaceutical companies in a partnership where we all come together to generate things like the natural history study and so forth. And this would be data that would be available to any pharmaceutical company to assess whether they have a potential therapy that's appropriate for a given ataxia or across multiple ataxias. And so I think the general word is that NAF is forming as a nucleation entity, bringing people together, and it like bringing the patients together and bringing the clinicians together, and now having the various pharmaceutical companies form a partnership with the ataxia community so that we can all move forward and figure it out as fast as possible what truly is a beneficial strategy in terms of a treatment to pursue.
Great. And Sue Hagen did want to let us know, or let everyone know that's attending today, that there is a clinical trial for MSA, which is a form of sporadic ataxia, and that can be found in the research section on NAF's website. So check there. And I'm looking here for any further questions. Kathy, you have a question. Are you there? Kathy Shaw? It looks like Kathy Shaw is having some audio issues, so we'll go to Susan Stenson. You're self-muted. If you want to unmute yourself to ask your question, if you're there.
Yes,
Hi, Susan.
I'm here. Harry,
Great.
Yes.
I want to ask you, I know there is a clinical trial right now for SCA 1, 2, 3, and maybe 6 and 7, but you talk about a clinical trial that's going to begin at the end of the year. And I'm wondering, I haven't heard that, and I'm wondering what it is. Is it a drug?
So the clinical trial that I was referring to stems off of some of the work we've done in the partnership or collaboration we have with Biogen and Ionis. And it's a general strategy, the same strategy they used, for example, for the clinical trial that's underway for Huntington's disease, another neurodegenerative disease that probably many of you have heard of. And this is a drug that targets expression of the gene that causes the disease in these various forms of ataxia. In terms of the SCA1, it's still a moving target, and we just hope it'll be in place by the end of the year. I think in terms of SCA2, the target is fixed, and it should be starting, if it hasn't already, very soon. But I think they are running that trial. There's a relationship between SCA2 and ALS, and there are, Lou Gehrig's disease, and there are many more patients with that disease. And so again, for a phase 1 safety trial, they can get the numbers of patients assembled very quickly so that they can test the safety of the drug. And then as they move into phase 2 and phase 3, bring in the patients with SCA2.
Okay, my daughter is doing the Trilazole clinical trial. How is that going?
I'm sorry, but I'm not a clinical investigator, so I'm very the specifics of any given trial. It's just sort of what I hear, and I just would rather not comment specifically on how a particular trial is going. I think perhaps maybe Sue can get you in contact with a clinical investigator who can give you more accurate information than I have at my disposal.
Okay, thank you, Harry.
Very sorry,
Thank
but
you.
next year maybe Sue can give this talk.
We should definitely have her on a webinar. Absolutely. Are you able to comment on the portable neuromodulation simulator PONS device? Somebody is asking about that.
No, I'm afraid, again, that's out of my area of expertise. And so
we'll get that person in touch with someone that can better answer that question.
Yeah, again, I'm very sorry.
Jody is asking a question regarding exclusion and exclusion criteria for clinical trials. She had a bad reaction to Rizulo, so that excluded her for a clinical trial for SCA2. She'd like to know if at any point she'd be able to participate in that trial or other trials because of that.
Well, again, I'm sorry, but this is out of my area of expertise. My understanding, though, is that the ASO for SCA2 is a completely different mechanism and a completely different type of drug than the rizulamide. And so I would encourage her to get in contact with the people who are overseeing that trial and ask them directly if she might be might qualify for the trial. But again, my understanding is that the safety trial is going to be done in patients with ALS, and then it's the phase 2 trial, phase 2, phase 3 trial that will bring in the ataxia patients.
Great. And last question today, will there be any trials regarding RNA interruption and SCA1 to your knowledge?
Yes, yeah. So this is now we're moving to the area of gene therapy and using what we call interfering RNA to have exactly the same effect on the SCA1 or 2 or so forth. And again, this is, as the earlier comments, the exciting thing about this strategy is, I know for example, Bev Davidson at Penn is making some really incredible progress on developing a gene therapy approach that has an off switch. And I think the exciting thing is that this would potentially be a therapeutic approach that could be administered much less often than the need to administer the ASOs. And certainly once we understand more about the potential side effects of reducing expression of the ataxin-1 or the SCA1 gene, then again, moving to a strategy like RNAi and the AAV vectors would be on the horizon. So it's certainly a very exciting area, and it potentially could be a major strategy.
Thanks, Harry, for that, and all of you for all your questions. A recording of today's webinar will be made available, and additional questions can be sent again to naf@ataxia.org. Thank you again to Harry Orr for all the great information that you shared with us today, and the NAF staff that contributed to today's webinar. We truly appreciate you all for joining us today, and certainly take a moment to complete the post-webinar survey. We'll look forward to having you all join us for the next webinar. Take care and be well.
So long.